Okay, welcome everyone. It's a pleasure to have Professor Rakesh Mishra here to deliver an institute lecture in the ACOM series. Professor Mishra did his PhD in, I think, organic chemistry in the University of Allahabad. And after some postdoctoral work in ISC and so on, he spent most of his career at CCMB Hyderabad, where he was the director from 2016 to 2021. And presently he is the director of Tata Institute of Genomics and Genetics and Society in Bangalore. It's a new private sector institution that was set up a few years ago. Professor Mishra's expertise is in a wide range of biology, but specifically in non-coding DNA and its function in Hox genes, in epigenetics, and he also does a lot of bioinformatic related work, primarily on Drosophila as a model organism, but on many other things also. He's a fellow of all of India's science academies, and he's apart from all of that an all-round nice person to know. He has been to this institute a few times before, and we are very happy to have him again.
Thank you, sir. Thank you for the nice introduction, and thank you for inviting me to this place. And it's my pleasure to talk to different kind of people who do mathematics. We generally talk to biologists, and so it was, and I had great time since morning listening to the PhD Viva and the interaction with many students and colleagues after that. So I really like that, and it's very refreshing for me. So what I'm going to talk about is something that we have been working actually for 10-15 years, which is something a little different from regular biochemical or genetic analysis of the kinds of things that we do. And as I go, you will see. So first I want to project that why we are interested in this kind of a problem, and then I will show you the way we establish this system and some I think interesting conclusions that we have come to, that we have some ideas about the structural basis of epigenetic cellular memory, which I will explain as we go. So this was just very simple statement that DNA is the genetic material that is inherited, but then there are things like transgenerational epigenetic inheritance which eventually depend on DNA but apparently look like they're free of DNA sequences. Then we are what is in our DNA, that is the phenotype, that each feature that we have is because of our DNA, and DNA is also genetic material, so that's why we continuously evolve. We means the living organisms. And then the third point is that the identical genome in each cell of our body, while each cell type has a specific epigenome. Epigenome means different functional version of the genome. So we have over 200 different kinds of cells. Therefore, the same identical genome does 200 different kinds of activity, and that's what attracts us. How amazing it is that the identical genome can be packaged differently. So that a subset of genes are exposed and certain subset of genes are hidden in a cell type. So imagine in a brain, all proteases have to be shut down, otherwise we will have after some time empty skull, because the proteases will digest our brain. But in the intestine, those proteases have to be active, otherwise we will starve of like a food. Even if you eat, you will not be able to digest the protein. And each cell has each cell has this protein. So the protein in a non-epithelial cell of digestive system can be a bomb for the body. So the body has to have a system, the cell has to have a system where certain genes are totally out of reach, and we believe that that happens because the way genome is packaged, that they are never seen by transcription machinery. And this process, once it is to achieve this process, is called embryonic development, where from single zygote, whether it's an egg of an animal or a seed of a plant, one day you have repeatedly you get the same result. So how this complex organisms achieve this fantastic amazing job. So as I said, that which means that genome is really packaged differently in different cells. Then of course, then what is the packaging code? And this complex packaging is not only maintained but remembered by the daughter cells during the cell division. So like some of our neuronal cells live for decades. So that means they have to keep the protease hidden from transcription machinery for those 70, 80, 90, 100 years. Right, so never let that express, otherwise the brain will get damaged. And the certain cell like in skin or liver cells, and many cells divide. Actually, most cells in our body keep getting replaced. So when they divide, they have to remember the same cell. The cell has to divide into 2 liver cell and nothing else. It cannot go back to zygote. It cannot go to undifferentiated. If it fails, the mitotic memory fails, the cell will either die or it will become cancerous. So it's a disaster if the memory is not maintained. And this is in a complex environment of this cartoon of interface nuclear, where there are tremendous amounts of compartmentalizations, which some of them we know, many of them we do not know. And this whole nucleus, when the mitotic cycle comes, has to get converted into 2, has to be divided into 2 things. So not only DNA that is replicated, but the entire structure of the nucleus is replicated. And again, then how this is achieved is the key. So how this is achieved, to answer this, we need to understand the structural framework and the molecular details of the genomic packaging in the nucleus. So there's 2 aspects of this. One is the genome itself has probably non-coding part of genome has acquired features that it can be folded into multiple possible ways, and with the help of developmental process and proteins, different pathways lead to different cell types. And then there has to be an environment. We all know to come and sit here for a talk, but there should be chair, there should be a stage, there should be projection, those kinds of things should be happen. And only when both are of same kind, then only it will be a functional. If the same thing be happen in a canteen, then we'll not have this, will not be able to have a good talk. And then here, if some other kind of people come who don't have no interest in this, then also it will not work. So that means the genomic features and the structural features of the nucleus have to change, and that is what I'm going to discuss today in a little bit of detail. So before that, as you see, from simple to complex organisms, when you go, you see the genome size increases dramatically. But it's not the number of genes that increase. So actually the percent coding genome kind of decreases. So outcome of evolutionary process is static number of genes, but more and more non-coding DNA. So much so that in humans, per gene you have 120 kb of DNA, while in lesser complex organism it's a few thousand only. And the complexity means number of cell types. So as I indicated, number of cell types in humans is it doesn't reach that far. So over 200, while simple organisms have lesser number of cell types, although number of genes may be very comparable. So more number of cell type means that many number of genomes or functional version of the genome, or the number of ways genome can be used or packaged. Right. So that's the big challenge that we address in the lab. But today I'm going to talk about the other side of the same process, that is, this is happening in the nucleus, and there has to be some basis, some framework, some architecture within which this genomic features are created, and that's how genome is packaged. So I'm going to introduce to you a biochemical preparation called nuclear matrix, so which is essentially a non-diffusible salt-resistant biochemical preparation which is believed to provide the structural basis of nuclear compartments and the processes. And it contains proteins which we call new-mat proteome, DNA which called matrix associated regions, and RNA which is a new-mat RNA. So DNA RNA is a different story. I'm going to focus today on the protein part of it, which we have been working for some time. So this is just to impress you that you take this is Drosophila nuclei in a culturing cell, and we take this, prepare the nuclei and then treat with the salt so that the histones are detached from this. So when the histones are extracted out, now the DNA cannot be contained within the nucleus. So what red thing in the middle you see is the I think this is not reaching. Okay, I'll not touch many things, otherwise the problem. Oh, this is here. Huh? Yeah. So you see in the red in the middle is actually lamin. So you start seeing not only the circle but meshwork of lamin that is revealed because the chromatin is oozed out, and the blue thing is the DNA which is oozing out, and the green thing is in fact the repeat, simple sequence repeat. We have hybridized it that because this experiment was done for something. So you can see the repeat also going and making those loops and coming out. So this means this nuclear matrix is within which the DNA is compacted, and if you disturb this compaction, this whole thing is not anymore viable, and nucleus is somewhere else and DNA is completely outside it. Most of it. So this is what we were interested in. This meshwork what you are seeing is because of the antibody against lamins. So we started to purify, make a preparation of this many years ago, how to prepare quality nuclear matrix. So there were procedure, I mean these people are working on this from 80s. But we standardized this procedure, we set the baseline for what is the quality control parameters, that when you begin what percent of DNA is retained, what percent of RNA is retained, and there are certain molecular markers like histone should not be there and lamin should be there and those kinds of things. So we standardize all this, and then we had the material to do DNA, RNA and protein analysis from this preparation, and we have done all of them. As I said, I'm going to focus only on protein part of it. So when you prepare this matrix, so actually you can go back and digest this matrix completely. If you add dissolve it completely. If you add 8 molar guanidinium chloride, the whole thing becomes solution. The matrix structure is lost, and then you can dialyze it to bring back the lower concentration of nuclear matrix. Now you can see the fibers get formed again. So these are the fibers we call as NuMat fibers, typically 10 micron, 10 nanometer in size. But if you treat the nuclear matrix with RNAs after renaturing or before renaturing, you don't see these fibers growing. And then of course we have made many variations of this, you can grow these fibers again and again. So which means the nuclear matrix that we are preparing, even if disturb it, you can make the fiber again, provided that you don't disturb the RNA component of it. So for this fiber, the RNA requirement is necessary. So we have so once we are sure about this preparation is good, it can be verified, and we have quality control methods. Then we started to do analysis like what kinds of proteins they are. So I will just go through just very quickly. So these proteins have now different kind of VGO terms and all. We can say that they are involved in signaling, they are a structural component, they are an interaction with DNA, RNA and chromatin, which was kind of expected. And we also did this at different developmental stages, and we see that lots of proteins change, nuclear matrix proteome changes depending upon the developmental stage, but about 50% of the proteins are constant, they don't change, right? So those we call them as a core NuMat proteome. That is same in all the cell types, no matter which cell type you take, but many others are cell type specific. So that was something interesting and unanticipated. So those were work in the flies. So we collaborated with Jyotsna Dhawan in our institute and did set up this mouse cell system, which is a very nice cell system of C2C12 cells, where they can be in non-dividing state called G0 or myoblast, or you can differentiate them into muscle fibers. And then we learned how to make a matrix from this. So this is cell alone, and now you can see this is a matrix prepared. So you can see the actin becomes visible and the lamin becomes visible and so on. And once we did that, then we of course did the analysis of what this proteome contains, and we got the same kind of things, like around 40% of the protein don't change no matter which is G0, MB or myotube, but other proteins reflect to some degree what kind of cell type it is. So which means this nuclear architecture part of this is fixed, which doesn't change. So we assume it like part is like walls of this and pillars which will be there in, or the bricks and all will be everywhere. But certain thing will depend upon what kind of functionality of that particular space is. So once we learned this much about the nuclear matrix proteome, we wanted to see, and we saw that the fly and the mouse systems are comparable. So we wanted to now compare that across the evolutionary line. What kind of proteins are beginning, they are always there, and some of the proteins that may be species specific, but we are more interested in coming to the core and the minimum nuclear matrix proteome which may have originated very early on. Essentially, we were interested in figuring out how this structure has originated at what time, if we can figure out that. So the way we did it was we used Drosophila and zebrafish system. We made nuclei and the nuclear matrix from the embryonic tissues, and so this nuclear matrix numat proteome was then first filtered and picked only what was common in mouse. Then we filtered so what was the nucleus from the human analysis, and then we reached a certain number, and then we looked at how many of these proteins have origin of prokaryotic and what all there in all eukaryotes, and some of them do not have origin of prokaryote and so on. So this is essentially the result. You don't have to see the number and those kind of things. It's just that there is a pattern, and the summary of this thing is that we found something like 80-90 protein families from the existing prokaryotic proteins were initially recruited for the nuclear architecture. So which means they have prokaryotic origin as well. And out of this, 36 of these proteins are constant in any eukaryote check. So of course we could check only for whose genome sequence is available, and the identity of these is based on the experimental data of mouse, fish, and fly and human, right? And then rest is based on homologue, and we are assuming that they will do similar work if they are doing from fly to human, same kind of things. So and then others out of say 40 of them which were not conserved everywhere, they were either lost or they have diverged to group specific specialization, and that's why we are missing them in our analysis. But in this process, we, in addition to these 30-40 proteins, there are about 200 additional proteins have come which are common in all eukaryotes, which we consider that they were recruited for various aspects of nuclear structure and function in the metazoans. So essentially what we are trying to, we are developing some kind of set of idea how the gradual accumulation of factors in the nuclear process and their inclusion in the nuclear compartment of the architecture began from, let's say, common ancestor to archaea to bacteria, and then when several rounds of endosymbiosis occurred, then so some of these proteins were then recruited for the architectural component, and eventually you get enough of them, about those 250 odd proteins, which can then provide a structural basis for compartmentalization in the nucleus, which as all of you know is so important for its functional complexity. So this is what we are doing is isolating the nuclei and then doing biochemistry and then making using instruments to identify proteins, DNA, RNA. And but this is not convenient if you want to do in vivo experiments. So we decided to do, it took many years, but we made a protocol for in situ preparation of nuclear matrix, and we can now do in many different kinds of tissues, but we began with Drosophila embryos. You want to ask something? Yeah. Yes. Yes.
But the transcription happens upon split stimulus, right? It doesn't happen on, like if I have a completely unfolded DNA, all the genes won't be expressing all that.
So some will. So there are some genes which are continuously expressed during the interphase because they are housekeeping genes, but many genes, you are right, they will express only upon a signal that the gene is open for business, but it will do only business when a customer comes. Otherwise, it will be waiting there. So there are both kinds. But
This constant background that the gene from that type.
yeah. So yes, so the housekeeping genes don't change. They have to be there. Every cell has to breathe, has to produce energy, and have enough to make packaging, remodeling. So many of those proteins are common. But there will be cell type specific, as I said, for intestinal epithelial cell, the protease will be expressed. In the brain cells, thinking genes will be expressed, or whatever those kind. So large number of, so there were several thousand genes are almost expressed in all cells, but then rest are cell type specific markers. Like
Things are not only on stimulus but also on stimulus.
yes, yeah, they are open in that. So so like MyoD and all, I showed two examples in the mouse cells. Though actually I just rushed through the first two pictures were actually the markers for the muscle cells. So that means they express only in those cells, and for other things they will be. Yeah. So here you see in the middle. So this is Ki67 and MyoD. It is not expressed in G0 cells. These are resting cells. These are, we call them muscle stem cells or satellite cells. When the cells decide to differentiate or to divide, then you have Ki67 and MyoD both expressing. But when so this is dividing cell. But when the cell differentiates, then the MyoD will continue to express, but not this one. So this has transient role to play. So this is how they are cell type specific regulation happen. I just gave this example, but there are hundreds of things. Yeah. So the idea here was to make the nuclear matrix preparation without isolating nuclei, without disturbing the tissue. So we take the entire embryo and make nuclear matrix in that. So I don't think you need to go through that process, and is also available in public domain. So but you can see that when we section these embryos and do EM pictures, we can see the fibers as one would see complex fibers being formed, and if you treat with RNAs, then they kind of start to break down, and you can now image these things like the same thing. You see lamine circle, but when you make nuclear matrix preparation, inner meshwork of the lamine becomes visible. Of course, this has no DNA because we have extracted the DNA histones out and so on. So once we established, then this was very interesting for us because this opened us the way to do many things. We can now use Drosophila genetics and imaging. These are powerful techniques to the whole thing I was talking about, right? So it became available. But so I'm going to show some of those pictures. And we were also, since with this during this guanidinium hydrochloride extraction, we were able to actually fractionate the matrix in different in terms of their diffusibility or their ability to solubilize into different layers. So you can see we have systematically this is something strange. No, it's relaunching the because I can't see in I hope this is the one. No. No, it's in the computer. Pen drive also I have, but Huh? Oh, so you're saying that. Yeah. Why this happened? No, okay. We would start. I don't know why did it. So maybe I'll touch less buttons here. So we can see now in what actually you see. What you see is that with depending upon 1 molar, 2 molar or whatever molar guanidinium chloride, you see different clusters of proteins coming. So something comes only in 1 molar, then some another set comes in 2 and 3, and this is what is plotted. And in fact, since we have the now in situ preparation, that is the biochemical preparation, and in situ we can match the similar things. You can see some of these things, like this is like, for example, a thing called polycomb. So you see there in the beginning up to 1 molar is there, but after this nuclear matrix and in 1 molar it's all gone, it's not there. While TRL, the Gaga factor, you can see some is there all the way up to the last and so on, and so they are different one. This is a fibrillary, which is nucleolus, bigger dots, and they are there till the end. So there is different solubility of these proteins, and which we are now analyzing that why they come in certain, they are more diffusible or less diffusible and so on. But what we were more interested in, which is what I'm going to talk about, is that this is a Drosophila embryo which I'm showing you, where the mitotic wave is going from the left to right. So you see in the behind more number of nuclei, and in the front toward the right end, those nuclei are yet to be dividing, and very end another wave of mitosis has happened. So then what this provides you is to all stages of cell cycle in one shot in the same animal, which we have not disturbed. We just had prepared a nuclear matrix, and now we can go and look at the nuclear architecture at different stages in the same animal, and that is what is the higher magnification, just to impress you here. And I will give you some pictures that you see here. Laminin structure, this is in the middle of the mitotic wave, where you see mitotic chromosome, lamin is gone, but this green thing what you are seeing is actually a protein that binds to is called boundary element associated factor 32. So you see, although lamin is gone, nucleus is disappeared, but this protein is still latching on. And these were the observation why I am going to tell you we got into mitotic memory of the thing. So this is the I wanted to impress you that now you can use the genetic tricks of Drosophila in context of nuclear matrix preparation. So you can do really genetic and in vivo experiment. And the example here is that you are seeing something called polytene chromosome. It's a long time, maybe 90s data from Lemley lab, which they made antibody against this beef protein. Beef has 2 isomers called beef 32A and B, and here you can see red and green are beef 32A and B, and you can see chromosomes are fully decorated. Most of them are yellow, very few are green, and many are red. So that means green and red means they are not overlapping, rest they are overlapping. When we so what we made 2 flies, and we tagged these proteins differently, one with MIC and one with flag, and make nuclear matrix in situ and ask if these proteins how they do, because in our proteomics we have seen this protein, that's why we were chasing it. So in the nuclei you see this is lamin, you see circle, and then this 32A chased with MIC, 32B, this is there, and this is the DOPI, and most of them will overlap, but still you can see some green, some red, as you see in the polytin chromosome. But what happens in the nuclear matrix preparation in situ, that lamine is there, but no 32A, only 32B, and of course no DNA and 32B. So that means although this protein has very little difference in them, only this part of this protein is different, otherwise they are same isoforms coming, they are basically splicing variants. So now this allows us to distinguish with such a sophistication that among the two isoforms, which one is a nuclear matrix component, which one is not, you can tell from here. So while we were doing this, I showed you mitotic wave, which is again seen here, that mitotic wave begins from here and is gone, and you can see here is the wave going, and this is undivided, they are preparing to divide. So when we started to image these as a nucleus, you can see these are mitotic stage, lamine is more or less gone, the blue is green is tubulin, which is making to, and in between is the chromosome, which will be then separated, and this is enlarged picture, the chromosome, the tubulin mitotic apparatus being built to remove. And when you do nuclear matrix preparation, now this is the same thing, but now we had made matrix, so you can see that lots of lamine is around there. It's not that any lamine has gone, it's everything is there, and because it's in nuclear matrix preparation, the DNA is now gone because we have digested that away. You see only tubulin preparation, no DNA, right? So you can see very nicely. So what I'm saying is that you can actually visualize what is diffusible and soluble and gone, and while the rest of the structure stays in place when you do new in situ preparation. So this we had done some time back, and here what the summary here is that when you do nuclear matrix preparation, even when the chromosomes are dividing and they are totally mitotic in inert, still you see nuclear matrix components, this is a mixed antibody still associated with the chromosome. So that means the nucleus is disturbed, actually dissolved completely, disorganized, removed, lamine is disappeared from there, but nuclear matrix is still, components of this still are remaining near the mitotic chromosome. So what are they doing? There is no compartmentalization, there is no any functional utility of this. So this is when we ask this question that there is cell type specific interphase, then it goes to mitosis, and then back to interphase with the same output. As I said, if you don't do it this way, then cell has to die. So what we did, as I have so far been telling you nuclear matrix preparation, we decided now, because this was old picture in a textbook actually made in 70s, that if you extract with the salt and all with mitotic chromosomes, you can get the what is called chromosome scaffold, right? So we decided to standardize this technique to a biochemical scale, that we can do proteomics of that and ask what proteins are present there. So we worked with the S2 cells of Drosophila and arrested in the mitotic stage, isolated such chromosomes and did the extraction and standardized again the mitotic chromosome preparation so that we can study the proteomics of the mitotic chromosome and compare it to the interphase nucleus. And this was the work done by Rahul and Rashmi. Rashmi has been the key person in doing all this nuclear matrix work from the beginning. So here what Rahul has done is compared the NuMat and mix mitotic chromosome scaffold proteome in Drosophila and this transmission of hallmarks of nuclear organization through mitosis. So we are asking if anything from the interphase is going through the mitosis, mitotic chromosomes in this process, and the result was very interesting. The features of nuclear architecture in the structural context of the NuMat are retained in the mitotic chromosomes and possibly play an important role in maintenance of the cell lineage specific transcriptional status during the cell division, and thereby serve as components of cellular memory. So that was the summary of this, and why we say this, because when we compared the two proteome of nuclear matrix and mitotic chromosome scaffold, about 2/3 of the proteins are common, right? There are very few mitotic chromosome specific proteins and very few, relatively few nuclear matrix specific proteins. So this means that much of the proteins stay put during the mitosis, they don't leave whichever part of the DNA they are associated with, and the proteins that are unique to NuMat or mix are low abundance ones. So that means they are non-structural parts, they are functional and they go and they will come again, they will be recruited. Chromatin remodeling complexes present abundantly in the NuMat as the core constituent but are absent in mix, which kinds of helps that NuMat is more happening place. No matter which cell type you are using, you have to remodel the chromatin and they are the staying in the their component of the nuclear matrix. But mitotic chromosome, there is nothing called transcription or any activity, so you don't need those proteins. They will, in fact, will be disturbance in the proper mitosis, so they are not as part of this. So those are some of the things that are not the part, but whatever is retained, we assume is the component which is related to cellular memory. So the way we are making this, proposing this model is that, so here is a picture you see, this is a cartoon of a mitotic interphase nucleus is preparing to divide and it becomes mitotic chromosome. And we have done proteomics of this, which is the mitotic chromosome scaffold or nuclear matrix, and compare that much of this is common, right? So here the idea is so you know the interphase chromosomes exist as territories, right? Chromosome territories. So we are saying that chromosome territory is squeezed into compact mitotic chromosomes, and this, which is built on the chromosome scaffold, is a compact and miniaturized NuMat inside, and most of the NuMat components stay with the mix. And so the idea is that the seed of to re-establish the next interphase are there already in mitotic chromosome and they are retained during this process. And so for instance, this is the cartoon of an interphase nucleus where you have chromatin organization in the interphase, like insulator binding proteins, cohesin, condensins, they are in different places. But in mitotic chromosome, they are retained in such a way that chromosome is compacted, but these things are retained. So when they will enter the next interphase, they will be able to attain the same kind of structural features which they were having in the mother cell. So here is the more complex model in which we have done many of these nuclear compartments whose seeds are intact in many places. So in between you have scaffold, but many other things are associated wherever they were, although they are not now being used. So the big circles have become small. That means small parts of this are there retained, and when this chromosome will decondense and become the interphase, you will see the same kind of interphase again. So essentially we are proposing a testable hypothesis. In a sense, now we have the whole system. You can, if I am saying that this is the responsible, you take that function out and you will see that cells will not be able to divide, or will divide and die, or lose its identity. So the mitotic chromosomes are miniaturized version of chromosomal territories of interphase state in the cell type, and much of the key components of the cell type specific constituents of the nuclear architecture are retained and used as seed for faithfully attaining the expression state comparable to that of the mother cell. And these features are likely to have close linked with embryonic development, because each cell type has its own unique NuMat components, and of course variety of epigenetic mechanisms. Because I was saying that once cell divides, it doesn't have to go through the entire developmental process. It should be able to replicate its nuclear architecture and genomic packaging so that the same cell type is retained, otherwise it's disastrous. So I will pause here. I will once so whatever message I had to say, I will I've already done. I will show 2 slides after in the end, but any question, anything, I'll be happy to. Yeah, please.
It will
replication happens, so now there are two copies of the genomic material, right? So,
Yes.
so the original volume and whatever of the bodies which were present as the extranuclear or extra genetic material.
Yeah.
So now you need two copies of them, right? Whatever clips and this and whatever structural organizational components. So you need double the amount now, and hence they have to be synthesized now extra, right?
So this is very interesting question, and that's why, if you remember, we made smaller dots in the mitotic chromosome,
Right.
bigger dot in interphase, because the same thing is divided into. So that's why we are calling that only the seeds. So
Right.
when they go to interphase and the transcription begins, this will be established. So actually, there is a larger context to this question. We do not know where rest of the proteins are, because we have not tagged and followed them. For example, nobody knows where the nuclear pore complex is sitting during mitosis, because you cannot establish very quickly nuclear pore complex when you have to establish from 1 to 2 nuclei, right? That's very complex structure. But they are not visible, so that means they are diffused somewhere, or so something which is not part of the chromosome but part of the nuclear envelope has to be around, and there also volume or the quantity is very, very important. So many of these things we do not know, that at what time before the division you have to have nucleus intact. Therefore, pore is complex, so that proteins are going in and out, is functional. But when it decides to divide, now it will suddenly require twice the number of pore complexes. So from where they are coming? So whether the same number is now divided into 2 and slowly more and more complexes are added, how fast is added? So we simply have not done those things, or nobody has done, so we don't know how these things happen. But here the idea we are saying is that when the nucleus is now decondensing the mitotic chromosome, which locus should go in the active compartment and which locus should go in the repressive compartment, the signatures or the seeds are there. So they will get attracted, they may get condensed or whatever, the phase separated in a way, because the seeds of those things are there, and then other things will come imported and make it whatever, larger or more stable.
Yeah, and I mean, ostensibly, since this is because part of epigenetic memory, so those new synthesized components will have to know where to go and sit, and that is not part of the genetic information, right?
Yeah, it's not, because the genetic information, that's why it's a continuous process. And so epigenetic elements are retained during the mitosis. That's what we are saying, that most of the interphase features are compacted within the mitotic chromosome. We do proteomics, it looks very similar, although nothing is happening there. So that means they are just retained so that they know where to go. It's like address written, and they will reach at that point.
Right.
They will migrate accordingly.
Thank you.
Nuclear matrix you have, if you can go back to that previous slide where you compared this interphase nucleus with the mitotic chromosomes. So all these nuclear body seeds are kind of planted, but there is some message hidden in how they are planted in such a way that it can assemble back into the same structure afterwards, and that part is
right. So essentially, you can say that now, so this dot is there, so that means this piece of DNA has to come to polycomb body, it has to be repressed.
Right.
So now many of those things assemble in one place, because they all have tags. So this and this and this, they will all come and converge here and make a polycomb body. This is our linear chromosome. I mean, now they are not condensed together, or they are not phase separated.
I realize also it's a cartoon, but so is there a possibility that the wrong blue dots will come together? And I mean, so these are things that you haven't.
No. So this is just a model which we're just able to publish recently. Actually,
right. Do you think one can learn something from something like yeast, a simpler system? Will it have anything like this? Like it has
each nuclei is so small that doing these things becomes difficult. Actually, staining becomes a problem. Otherwise, that's the ideal system, because every gene you can every gene is tagged and available the library, and I have access to all those. But yeah, we haven't done that because this imaging to this resolution becomes very challenging indeed. But I think now, since we know what to do, one can do it specifically. Yeah.
Hi, yeah, so this is a much more basic question, going back to what you said about, selective expression of genes depending on cell type. So my understanding as a theoretician was, based on the Kaufman model of the, 70s, that essentially, the genes regulating each other form a network, and there are certain patterns of expression activity which depending on the initial condition would mark various cell types. So in this picture, of course, there is no role to be played by, in some sense, the structure. Because here you're just saying that, if you connect various genes according to who is regulating who, or who is positively regulating who is negatively regulating who. So these cell type specific expression patterns just appear as attractors of the network dynamics. So how would you say that your structural pictures, let's say, modifies this more networky picture?
No, it doesn't necessarily modify. See, any network is a theoretical concept. Now, whether they come physically and get attached, that's how they are network, or they are communicating through some feedback or some other mechanism that in the end, somehow whatever you are saying is network has to touch each other in some ways, right? And here, since there are no radio signaling system, it has to through molecules. So molecule is made and it affects this one, then goes this, and then eventually lands there. So that's how we look at that. If thing is happening in a physical world, at some time they have to physically find one another through a molecule or through direct interactions. So what here I'm talking is that there is a framework in which genome is accommodated. Now this, and we also know that different cell types have a constraint that this gene has to be interior, hidden in the territory, so that transcription machinery never sees this. And here are the genes that are on the periphery of the territory, so that transcription machinery sees them, and whether they are constantly or stimulus based transcription happening, they are available for transcription. And whatever is transcribed goes through the pore and protein is made, and then it comes back and does the job. So this organization of hiding what you don't want to expose in that cell and making available what you want to be seen on the surface, and that is a simpler picture. In fact, these are more condensation or clustering of the loci happens, because we were talking before, in each cell you have only maybe few hundred places where transcription is taking place in the nucleus. Those are called transcription factories or transcription foci, maybe 500 in a human cell. But several thousand genes are being transcribed. So that means each cluster, many of genes have to come in one place in the one space in the nucleus. So that's what we were discussing. So here, let's say transcription assembly. So here is a transcription happening here and here and here. So this is happening, but then transcription assembly, here mark is there. So these few of them will come and assemble in this part. So something which was bringing them together, when these chromatin, they may be coming from different chromosomes even. When they separated, they retained a little pinch of this along with them, so that when the decondensation of this chromosome happens, they will come back here again. And of course, there is in the middle the scaffold on which this DNA is attached. So even that scaffold will reorganize itself and become nuclear matrix here. So that means the entire structure of the scaffold will swell and differentially go either on the surface or stay inside, depending upon what kind of seed are attached to that. So that is the model we are at the moment talking about. So that's why we are saying this is some structural framework of mitotic memory. So we can now at least put something, and this is testable because now we are talking which are those proteins which are involved. So you can now mutate and check them. Yeah. Rohit? Go ahead.
So it's a very, I just observed this picture, and so there are multiple small portions of nucleolus on your seeds for nuclear bodies retained on mitotic chromosome.
Yes.
And there are other polycomb bodies, schedule bodies, but once they're formed again, you have small portions of the rest of them in other places, but nucleolus is one, right? So is that a constraint that's satisfied all the time?
Yeah. Actually, that's very nice question. I should have taken that as an example. So you know that the genes that come and the ribosomal genes are many chromosomes. They all have to come to one nucleolus, and many cells have only one nucleus. We can create condition when nucleolus gets fragmented. So in fact, we have identified RNA which are part of nuclear matrix. When we mutate that RNA, then the actually embryo, the animal dies. But one thing that we observe is the nucleolus is fragmented. We see 3, 4, 5 nucleolus in the nucleus. So that means we have broken the material or the framework on which the nucleolus was sitting. So it's now disturbing and becoming multiple copies. But coming to what you were saying now, wherever the genes for the ribosomal RNA genes are there, that is where some of this fibrillary, I showed you in one of the staining, that fibrillary is even after making nuclear matrix preparation in situ and even washing it with the high concentration of guanidinium chloride, this is still around. So that means they are still in various different places sticking, and they can come back once you have the reorganization.
Thanks.
Any other questions? Yeah, you can carry on.
Okay, so I'll just show 2 slides just to show that I talked about nuclear matrix story. In the lab, we do few other things. Okay, it went off, so I'm not going to again. So we have few other interests in the lab which I was trying to show. One is how anterior posterior body axis is defined in animals, and we use Drosophila as a model for that. And we have other interest in looking at the function of non-coding part of the genome, which we talked about in the beginning, and we are particularly interested in simple sequence repeats and their functional relevance, and now we have gone more detail into what proteins bind to them and so on. And then we are interested in something called transgeneration inheritance, which means if you give a biotic or abiotic stress, what we observe is through male germline, this stress is transmitted, the effects of this is transmitted to several generations in the flies, and we have started dissection of that. The genetic dissection of this transgeneration inheritance, it may sound funny, but in certain mutant background it doesn't happen. So that means there is a genetic system built for the transgeneration inheritance, and we are now looking at what are the molecular players in this process. So that is my activity in CCMB, and I'll just take 1 minute to advertise about Tata Institute for Genetics and Society, where I'm affiliated also now in Bangalore. We are in Bangalore Life Science Cluster. We are actually located inside INSTEM and NCBS building, where we have lab space. And our aim is to, and this is the institute funded by Tata Trust, and aim is to use cutting edge science to solve societal problems. And we have very specific program which are directly linked to societal outcomes. One is infectious disease, where we are implementing surveillance of pathogens in wastewater, so environmental surveillance, and we are very much connecting for leading partners in the One Health system of Bangalore, and we are making a model of small city, big city, so that it can be implemented in 2 years in the rest of the country. And we have a big program on vector control. We have probably one of the best insectary in the world, which can do modifications of insects, mosquito. We make transgenic mosquitoes of various kinds, and idea is to how to make environmental friendly methods to control vectors without going and destroying all the insects. Can be designed the ecosystem for controlling mosquito where we live, and we don't have to go and ruin the whole environment. So that's our infectious disease program, and we have a rare genetic disease program where we try to work on diagnosis, interventions, and outreach programs about rare genetic disorders that are unique ones in this country, where there is no other company or other institutions involved. There are large number of such diseases which go unattended. So we have set up actually a cell-based therapy and mRNA therapeutic platforms to address these at a cost which will be orders of magnitude lower than what is currently available. The current treatments of these things are just simply unaffordable. They cost, some of you may have read, 16 to 20 crore for one-time treatment, or 50 to 80 lakh per year. So it's just simply not possible. So, but we know that this can be brought to really like a normal drug. So that's one thing we are doing. And we are also doing something in crop improvement, where we are using genome editing technology to make climate resilient and nutrition, better nutritional crops. Just summary. So I'll not drag it more, but that's my new business happening, and my lab is in CCMB. Thank you for your attention.